Okay, so now we're going to get into translation. So translation uses the nucleotides in the messenger RNA to code for different proteins. Hold on a second. This is a codon table that shows every single codon that could be seen in your messenger RNA. 61 of these are called sense codons and code for amino acids. 3, UAA, UAG, and UGA code for protein synthesis to stop. And so these are called nonsense codes. If you notice, every single amino acid has 2 or more codons associated with it other than AUG methionine. Methionine here is the start codon. It starts all protein synthesis. It's always the start codon. And one of these 3 will be the stop codon. The other nucleotides between the AUG and the UAA per se are going to code for amino acids. We read our DNA or our RNA 3 nucleotides at a time, and that's considered a codon. And it's how those nucleotides sit together that will determine what amino acids are coded for. The fact that we have multiple codes for the same exact amino acid, and here you see 4, 5, 6 codes for leucine. Over here you have 4 codes for proline. Here you have 2 codes for tyrosine. That basically helps us to reduce the likelihood of mutations occurring. And this is discussed in the wobble hypothesis, which basically states that the first nucleotide and then the second nucleotide are the most important nucleotides. The third one is least important, so it's less important than the other 2. So you can have a GGU here or a GGC here, and they both are going to carry glycine. The transfer RNAs, the same transfer RNA can carry glycine and bind to different nucleotide sequences so long as the first and second nucleotides are the same. This reduces the number of transfer RNAs needed, and it also reduces the likelihood of mutations occurring. This is from a different textbook. I just like this figure, and it discusses the wobble hypothesis well. So now we're going to get into how translation occurs. The region to be translated is going to be designated by the RNA nucleotides. You always start at a region called the start codon, and you end at a region called the stop codon. The ribosome will bind at that start codon, and there's a large and small subunit that binds, allowing the different transfer RNAs to come in and bring along with them their amino acid. So here is a ribosome, the 50 subunit, 50S unit, and 30S unit right here, and with them, or they bind at that start codon region, and then amino acids that are held on these transfer RNAs are going to move in, and we're just going to continuously add amino acids to the growing polypeptide. So this is going to be produced in a 5' to 3' direction, just like everything is. Transfer RNAs have a unique shape. I always make them look kind of like a T. and they basically have one region that's really important that binds to the messenger RNA, and then they have another region that holds an amino acid. So they'll hold whatever amino acid that is coded for in the messenger RNA, and then they carry the anticodon that allows them to bind to the proper region. So this is what it really looks like, but I always draw mine kind of like a T when I draw it. You'll see. And so translation in prokaryotic organisms can actually occur or start occurring prior to transcription finishing. In eukaryotes, this can't happen because in eukaryotes, transcription occurs in the nucleus of the cell, and ribosomes are not going to be able to start translating until the messenger RNA enters the cytoplasm and can bind to the ribosomes. Oh, I will also mention, you can see multiple ribosomes here producing proteins. Prokaryotes can have multiple ribosomes reading and producing different proteins because this can code for more than one protein. And we can produce a lot of the same proteins as well as the ribosome moves along the transcript. So here's what your RNA looks like. This is the 50S unit, and there's your 30S unit. It binds on the messenger RNA. You have this E site, which I don't really talk about. The P and the A site are the regions I typically talk about. The E site is kind of like the exit site. The first transfer RNA that is going to bind will be methionine. Methionine comes along and it has the anticodon that allows it to bind. So this is a transfer RNA. Here's your methionine. And then the next nucleotide or the next amino acid that's going to come along in its transfer RNA is going to have a GGC anticodon because it's going to bind here. And so here's GGC on that transfer RNA, and here we have proline. So now the entire ribosome is going to move one codon sequence. This methionine is going to stay in the first spot in this P site, and this transfer RNA will move to the P site. And so we're going to actually make a bond between this methionine and this proline. That bond is called a peptide bond. So there's your peptide bond forming. And now this original transfer RNA that carried the methionine enters the P or the E site and is able to exit. This one now is in the P site, and now we have the next one coming along carrying the AUG and tyrosine. So it binds here, and then we're going to shift everything again, and we're going to produce a peptide bond between proline and tyrosine. And now we have another one coming along. This occurs until we hit a stop code. Once we hit that stop code, that's going to end or terminate translation. The ribosome will come off. The amino acid sequence, this right here, this polypeptide, is going to be able to fold up into its functional form. The ribosome can be used again. The transfer RNAs are going to go off and pick up new amino acids, and the messenger RNA can be reused if we need it to. In eukaryotes, this is a little different because in eukaryotic DNA, we don't have 100% of our DNA coding for proteins. So the first thing we have to do is we have to get rid of the regions of our DNA that didn't code for protein that we coded. Then we're going to add a guanine cap and a poly A tail. This is going to protect the messenger RNA, and it's also going to allow translation to work a little bit more effectively. The poly A tail protects the DNA or the messenger RNA from being broken down. So when we produce that original transcript, we have what we call immature RNA, messenger RNA. DNA in eukaryotes contains the expressed portions, which are the regions that code for a protein, but in between we have regions that don't code for the protein. So these are called introns. We have to get rid of these introns and splice together the exons. So we cut out the introns, we splice together these exons, we add a guanine cap that protects our DNA or our messenger RNA, we add a poly A tail that protects our messenger RNA and allows translation to occur more effectively. And then once we have done all that, we now have a mature messenger RNA that can leave the nucleus and enter the cytoplasm.
Gene information is processed differently in prokaryotes and eukaryotes because prokaryotes lack a nuclear messenger RNA, mRNA, can associate with ribosomes. Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Eukaryotic mRNAs are modified before they are translated.
Their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single
All right, so I'm going to
protein.
stop here, and then we'll get into how organisms can modify the protein synthesis that they have.